Hello, and a very warm welcome to today's BioProcessing International Digital Week webinar on ligand discovery strategies for improved custom purification solutions, brought to you by Astrea Bioseparations. I'm Daniela Steele, Senior Product Manager at Astrea Bioseparations, and I'll be introducing our speaker for today. But before I do, I'd like to encourage you to send your questions for our Q&A session at the end of the webinar by using the chat function at the bottom of your screens. We will try to answer as many of these as we can, but if we run out of time, we will respond to you directly. So now over to our speaker for today, Eric Bugglin-Borer. Thank you.
Thanks, Daniela. Welcome, everybody. As Daniela said, I'm going to be talking about our ligand discovery strategies for custom purification solutions. I've been working with chromatography. My background is in chromatography, and with Astrea, I help support working with customers to develop their projects. Today, we're going to focus on a couple main things. First is understanding the ligand discovery process for two approaches that we use. The first that I'm going to talk about are the synthetic ligand libraries, and the second are aphamer ligands, which I'll talk about both of those, the relative advantages of each when targeting your specific molecule. At Astrea, our story is one of discovery, development, and delivery of unique solutions to our customers. The custom ligand discovery process I walk you through today follows a similar path to what our R&D walks through with our customers and with internal development of new off-the-shelf products designed for purpose. While the focus will be discovery, which is the first step, I'd like to touch on a couple of the things that we do after discovery. We can develop custom resins. We can scale up and produce 1,000-liter batches. This is all done in our clean room environment. And we can provide the regulatory support so that you can use these resins in GMP processes. Interestingly, Astrea Bioseparations is the only bioseparations company to offer complete control and oversight over the entire design and manufacture of your product. How? We manufacture our own base bead from the raw materials. We can help you discover your ideal ligand for your separation. We create custom adsorbent by attaching your unique ligand to that base bead. We produce custom products at manufacturing scale, and we can also custom pack these resins in GMP-ready columns for you. In an environment plagued by disrupted supply chains, we help ensure security of supply. And this, I think, has been very important, especially as lead times are so long right now. Kind of talking a little bit more about security supply. We have two sites where we manufacture our Puribead. That's our base bead. It's a green process that we use, so we don't need to use any organic solvents when we're making our bead. And also, we have very tight control through our proprietary process of the bead size, and this allows us to reduce waste. So, not only do you get security supply, you're also helping the environment. Additionally, we have on-site buffer prep, we have plenty of storage space, and we have an effluent treatment plant. So we see the process through from beginning to end. As far as quality control is concerned, we have the ability to develop GMP-ready resins and columns. We're ISO 9001 certified, and we have ISO 8 and ISO 7 controlled environment production areas. Now, getting into the background of affinity chromatography. The resins that we're going to be talking about, it's going to fall under the category of affinity chromatography. And there are several challenges when it comes to finding the right ligand. You need something that's safe, scalable, and cost-effective. We want to be able to selectively capture from a complex feedstock, separate our target from any process-related impurities and also separate from product-related impurities. The clear example here is protein A, but not every molecule has an FC region, and so that's why we have this, this custom service. So looking at the ligand discovery process, the ligand design is a very important part of the affinity absorbent development and to help us meet your challenges. In many ways, it shares similarities to the discovery of new small molecule drugs, including identification of a selection of compounds, primary screening, and identification of lead compounds, and structure-activity relationship studies. It's followed by characterization, screening, and design of the target ligand, and then secondary analysis. However, there are some additional constraints that we have to deal with. And these include a requirement for the affinity ligand to be attached to a solid support and oriented correctly in order to interact with the target protein's binding site. In addition, we can't bind our target too tightly because we need to be able to release that after we've washed everything else away. So there's a balance to be achieved between affinity and avidity. The goal is to get a ligand that binds a target protein with high selectivity, but that can also be desorbed with relatively simple solutions or additives without adversely affecting the target protein. And here you can see what we're looking at when we're trying to select an affinity ligand in our manufacturing process. As I mentioned, there's a high selectivity, reversible binding. We're also looking at binding capacity, robustness, resistance to fouling. There are lots of different options when we're looking at affinity ligands. So there are natural binding ligands, antibodies, engineered proteins, peptides, and small synthetic molecule ligands, which is one of the things we're going to be talking about today. For use in GMP process in the manufacture of a human therapeutic, we need to be looking at all the adsorbent components. We can't have anything that's toxic or mutagenic. We're actually going to also be looking at cost of the ligand. That's a major consideration. And some ligands, as you know, can be quite costly, especially if you have to manufacture a protein or antibody that you're going to be putting onto your base bead. We also have to take into consideration ease of production, security of the raw material supply chain. It's not uncommon for at large scale, say 100 liters, to require a kilogram of ligand. So we're looking at how easily can we actually get this raw material. So the chemistry that we prefer to use for the small molecules is a triazine ligand. And these triazine ligands bring many advantages and check off each of the things that we're looking at. So they can be designed for selectivity and avidity. They are small and synthetic, so that allows for scalability, that allows it to be much more easy to source. They're highly stable. A lot of our resins we can hit with sodium hydroxide to do CIP. Long lifetimes, so we're not degrading a protein, we're using a small molecule. And then they're non-toxic, non-mutagenic, and they're well established in regulated environments. There are many routes to discovery of new synthetic affinity ligands. The very easiest is you already have a ligand and you just want that, you just want us to attach that to a bead and manufacture that. We can do that. But if we have to go into discovery, then the first question we're going to ask is do we know the target protein structure? Is that known and available? And if the answer is yes, then the next question is, well, do we know the ligand binding site? And if we do, then we can use ligand docking and high throughput virtual screening to determine potential binding ligands. Ligand docking means that we can take select ligands from our virtual library, and the next slide is actually going to be showing this a little bit, but we can take select ligands from our virtual library and place them in the binding site of the target molecule and evaluate the fit of the ligand with the binding site. And we're going to be looking at binding energy. If we do know the structure, but we don't know about the binding sites, then we have libraries available where we can do extensive screening. And here we can use binding site determination or blind docking to identify ligands suitable for the target protein. This is where the virtual ligand library is screened in silico against the protein to identify ligands that have an affinity to the binding protein site. Now, what if we don't know about our protein structure? Well, we might know about the ligands or inhibitors. And if we do know that, then we can search for ligands with structural similarities and design our molecule that way. There are, of course, cases where we don't have structural information about the target molecule. We don't know about ligands or inhibitors yet. And so we have a ligand discovery approach where we perform systematic high throughput screening of a selection of libraries and identify a hit through screening of a more discrete but diverse library. We're looking for a region of the protein that would make a convenient site for the binding of a ligand. It doesn't actually have to be part of the natural biological function. It just has to be a part of the target that will bind to our molecule. And I'm going to talk a little bit more about this later. So I touched a little bit on virtual library screening, and this is an example of a virtual screen. In this case, we wanted a ligand that could be used for multiple isoforms of the target protein. So we started by determining common clefts accessible to the ligands. The protein scanned for putative binding sites, and then the ligand structures are virtually docked to these binding sites, and the binding energy is determined. When selecting possible candidates, it's important to consider the ligand orientation because we need to orient the ligand correctly when we're binding it to our bead. And so this is where we start to look into the influence that spacer arms might have on binding. Here are two examples of the output of the in silico screen. And this is looking at the binding fit of target molecules in the binding space. So you can kind of see on the proteins, we're looking at the same binding space, and these two ligands interact differently, but both had high scores for affinity to this target. Based on the structures from the virtual screenings, we can then design a library of triazine-based compounds that explores the chemical space around the structures selected. So we might not necessarily be using either of these molecules, but we might be trying different ways of mimicking their interactions with that binding site. And here we look kind of into the construction of the triazine ligand libraries. When we use these for screening, it's made with combinatorial chemistry. What that means is that the immobilized triazine ligand, it can be modified at 2 points. And that can be used to create a bifunctional two-dimensional ligand. And with access to thousands of amine compounds, we have the ability to make a wide array. we can rapidly develop very large and highly diverse libraries with novel binding functions. And the large size and diversity of the ligand libraries consequently increases the probability of a successful discovery program. At this stage, the ligands are made using solid phase synthesis, where the ligands are attached to chromatography beads. And this allows rapid screening of representative examples of our affinity adsorbents. So we're not just filling up 96 wells with ligand and trying to see what the binding is like. We're actually making lots of resins, and we're screening those resins. And so, we do this in 96 well plate, and these end up being discrete microcolumns. So they're a quarter of a mil each, and each one has a different ligand. We can create multiple libraries with a wide range of chemical compounds and a wide range of properties, and screen against our target molecule or against an impurity. This is kind of three views that bring us through this process. So in the upper left we kind of show the microcolumn plates, and they're used not only for ligand binding, but also investigate many other properties of the ligand, such as binding conditions, the effectiveness of different washing conditions, elution buffers, as well as the recovery at each step. And this allows us to gather large amounts of information early on in the discovery process. As much as possible, we like to do the screening with real feedstock. We want this to be done with real life conditions so that when you're using this, it works as desired. We don't just want to make something that's good in theory. We want to develop something that's excellent in practice. So going down to our second picture, when we're screening, we come up with heat maps. We produce these heat maps where the red indicates potential ligand candidates. This is high binding species. And we take this a step further by making this scatter diagram. This scatter diagram shows actual screening data from elution fractions from the microcolumn plates with protein purity on the vertical axis plotted against the concentration of the target protein on our horizontal axis. So what you can see is down in this corner, we have very low binding of our target protein. We also have very low binding of total protein. These are not going to be interesting to us because they clearly don't bind much of anything. Data points up here might seem interesting because we have very high binding of our target protein. Unfortunately, we also have just high total protein binding. And so these are somewhat promiscuous, and they're not giving us the specificity that we want. They're basically binding everything. So what we're looking at, the molecules of interest are in this circle where we have high target protein binding and relatively low total protein binding. So these are areas where we have selective binding of our ligand to the entity that we're interested in. Some people might be wondering, well, how long does this whole process take? Using these techniques, binding ligands can be identified in 12 to 16 weeks. And this is our screening strategy for general ligand libraries. Once potential ligands have been identified, their structure can be assessed and further ligands can be produced with a similar structural design in an iterative process until suitable ligands have been identified that fulfill our purification goals. After several suitable ligands have been identified, the adsorbent development stage can start. And this screening process is pretty quick. At this stage, we're aiming to provide target ligands attached to a chromatography support for verification of the purification performance. That gets us through our discovery portion. And so we would go to our development step after this. And that is a topic for a future presentation. I'm going to move on now to talking about our aphamer ligands. This is a technology that was developed by Avacta, and we've now partnered with them, and we're able to offer custom ligand development using this unique platform. Of course, you're wondering, what are aphamer ligands? This shows the size and shape of the aphamer in a ribbon diagram. They're based on Stephan A, which is a naturally occurring human protease inhibitor. Aphamers bind target molecules with similar specificity and affinity to that of antibodies. Some of the advantages are that they're small. They're around 10 kilodaltons, and the aphamer protein scaffold is biologically inert. This scaffold is based on a cystatin fold and permits the presentation of two variable binding loops, which we've highlighted. These present highly specific, high affinity binding surfaces for specific interactions with a wide range of targets. You'll also see in here that there's no natural cysteines or post-translational modifications in here. This ends up being important in how we're able to orient these when we're binding our aphemers to our bead. And I'll talk a little bit more about that too. So aphemers, as I mentioned, they have high specific binding properties. There are several other features that are very appealing for bioprocessing. So the screening methodology used allows the engineering of specificity and capacity during the selection process. In addition, the aphamer reagents are easy to manufacture in bacterial expression systems such as E. coli. As it's a small protein with no modifications or disulfide bridges, we're able to eliminate the batch-to-batch variation issues that often plague antibodies. And of course, the ligand has to be attached to the surface. And in the case of aphemers, this is relatively straightforward to control. What we can do is we can insert a cysteine anywhere in here in order to orient the aphamer when we're immobilizing it on our base bead. And due to its biophysical stability over a wide range of pH conditions, we're able to get a high level of reproducibility and the ability to endure cleaning routines. And as I touched on a little bit, how we immobilize the aphamer to support matrices. Both the scaffold and randomized binding loops are engineered to lack cysteines. And so when we introduce one, just using basic molecular biology methods, we can allow the protein to be immobilized in a defined orientation. And we do this using, as you can see, thiol chemistry. So getting into the discovery process, it's a little bit different with the aphemers than it was with our mimetic ligands. And this is a schematic. It shows the 8 separate stages. And the first step is the quality check the sample material that represents the target protein for this discovery process. During this target protein QC, incoming targets are tested for purity, biotinylated, tested for biotinylation levels, and this actually only requires small amounts of target protein, typically under 100 micrograms. Next step you'll see is phage display. And so phage display is well established and robust. It's a reliable in vitro selection method, ideal for the generation of affinity reagents. The first step, we construct a library. We then incubate that library with the target protein that's been immobilized to a surface, such as a microtiter plate. The surface is washed to remove non-specific binders, and then we elute the binders. The eluted phase population is then amplified, and we call this whole process biopanning, and we repeat it 2 to 3 times. This allows a very flexible approach to examine different criteria. For example, it's possible to select for affamers that compete against related targets. We can also vary the format of the target between selections. It's also possible to make the selection in buffer match to the end use, or in the presence or absence of associated ligands, as well as screening mutant versus wild type. So we can come up with the constraints of our separation, and we can test against those. Next up is the cloning and formatting stage, where the phage display output is mini prepped and affamer population is cloned into an expression plasmid. The primary screen allows the selection of multiple hits, again, only using low target consumption. A little more in depth with the primary screening is this is a bead-based assay, and so is more representative of the final adsorbent. So this is kind of like with the mimetic ligand, where we made the libraries on the beads and tested those. We're doing that here with the affamers. The advantage of using the IQ technology is that it uses 100-fold less target than a standard ELISA. These hits, again, are sequenced, and the unique sequences are bulk expressed, and then we move into validation. For validation, we use surface plasmon resonance imaging, which enables a large number of different label-free molecular reactions to be monitored simultaneously and in parallel on a single biochip. We can monitor the interactions between affamers and the target protein and look at multiple factors such as affinity, specificity, and elution conditions, and we can compare and rank based on these criteria. Again, the time for this overall screening process takes around 12 to 14 weeks, but the success rate for this is greater than 90%. So very high success rate when we're doing this screening. This is a very specific ligand that we worked on developing. So we were targeting human factor 9, and which is a treatment used in hemophilia. The target for phage display selection was factor 9, and then we had deselection targets for phage display, factor 2 and factor 7. The phage display was carried out using the standard method with a double loop library against a biatenylated factor 9, following pre-panning with biatenylated factor 2 and factor 7. And then we used unmodified factor 2 and factor 7 for competition during pans 2 and 3. We ended up selecting 167 clones from the pan 3 pool and followed the subcloning into expression vector clones. The affamers were purified from small scale expression cultures, and all the clones were assayed in our primary screen in 2 configurations. We ended up with 42 positive clones that we sequenced, and this ended up yielding 5 unique sequences for characterization. All 5 of these affamers were subcloned, for addition of a C-terminal cysteine, which was expressed, purified, and characterized. And here you can see the results that we got with our 5 adsorbents. So all 5 were subcloned for addition to that C-terminal cysteine, and we used each of them to do the purification. And you can kind of see here that number 3 was the best one. So that ended up being the one that we selected for the adsorbent development, and that became the final product. Now, you may be wondering, can I only do it on your base matrix? And the answer is no. We can, I mean, that's our preferred base matrix, but we're able to immobilize your custom ligand on whatever base matrix you want to use. The base matrix choice is going to depend on several factors, including performance, chemical compatibility, cleaning and reuse, the number of cycles, the cost that we're targeting, and so forth. These are all things that we're going to be defining before the project starts. And so to start wrapping up, we talked about the Triazine ligand, and we talked about the aphamer ligands. And you might be wondering, well, how do I choose one versus the other? And each has its own benefits. And so this is a thing where we can work together to figure out which of these is going to be the best fit for you. Ligand discovery, of course, is only the start of the journey. As I mentioned earlier, the next steps are adsorbent development, followed by manufacturing and scale up, both of which are part of the complete custom service offered by Estrella. These will be the subject of future talks. If you'd like to hear more in the meantime, feel free to reach out to us. We're happy to talk about it. This is a thing that we really enjoy doing. Thank you for joining us, and I'll turn it back to Daniela.
Thank you, Eric. That was a really nice overview of the ligand discovery process. We've had a number of questions come through already. I would like to say to everyone who's listening, please carry on sending your questions in. So, Eric, maybe I can pass over the first question to you. This is one that I think you can answer quite quickly, actually. Can affinity chromatography change the chemical nature of my target?
No. So the way affinity chromatography works is you're getting an interaction, but there's not a there's nothing that's changing your target.
Okay, great. And then I think you actually answered this during the talk, but I just mention it one more time here. Can these ligands be used in other formats, such as plates or on membranes?
Yeah. So as I mentioned, our go-to is using our pure bead, but we can also use membranes or other resin bases.
Great, thanks. I think we've got time for just one last question. Again, I think you did actually mention this quite extensively, but just to wrap up, I suppose, what factors would affect my decision to use a synthetic ligand versus an aphamer?
Okay, yeah, I can just go back real quick. So I can go a little bit more in depth on this. So the triazine ligands, there's a very highly diverse library of those. They're highly stable and non-toxic, and they're very straightforward to scale up. So it tends to be a little less expensive to go this way, and there's an excellent track record in regulated downstream processes. Aphemers are going to give you more selectivity, so it's just an exquisite selectivity. And then you're still able to get that chemical and thermal stability. and get that broad tolerance. This is a thing where when you do come to us with a custom project, we can explore both of these avenues and determine which is going to be a better fit.
Great. Thank you very much, Eric. It's been very nice to hear from you. And so if we didn't manage to get to your question today, Eric and I will make sure that we come back to you with answers. But thank you very much, everyone, for giving us your time, and we'll be in touch. Thank you.